Okay. I want to go over the algae lab data with you and kind of talk about what it means, and then show you what you're going to do to graph it. Some of you may already have gone in and started tinkering with that, and that's another lab grade. It's a big one. So we'll do that together. Yeah, I know, Sydney. Yeah, I know that happened. That's the thing, is that this virus can attack anybody, can't it? And definitely when we start the cell unit, we should talk about viruses. We should definitely talk about viruses and what their structure is like and how they're different from our body cells and what that's all about, because I think that's that you guys would really, really need to know about. I'm sorry, I've got to keep finishing this here before I can really get started with you. But you know, since you're going to be retaking the test in a few minutes, I would get biochemistry notes out. Okay, because you're going to be allowed to use them again. So kind of get ready to take this again. Although I have a feeling the second time you see these questions, it's going to be probably easier than the first. Yeah, that's what I'm hoping too, Zishen, is that vaccine comes along. That's another thing we should talk about. What exactly are these vaccines and what's in them, right? That would be a really good thing for you guys to know about. We'll definitely talk about that. So you guys realize that we don't have class together on Thursday because it's the PSAT day, right? So this is the only class together this week. Yes, Youssef, what is it?
What's going on PSAT day?
Oh, PSAT day is it's an exam. It's a national exam that's given.
Are the year 11s are doing, right?
Yes.
Like,
I think there's a national population that they're giving it to.
Okay, I got it.
Yeah. Good question. Yeah, okay, let's not talk about politics. Let's just stick with science. Oh, well, I mean, that's up to you. Everybody can take it. I mean, unless you got 100, there's no reason. Right? If you got 100 on it, but I don't think anybody got 100 on it. 25 questions. There are a couple of people who got close, but we don't have any hundreds. So everybody, go ahead and take it. If you only got one question wrong, they'll fix it, right? Okay. So yeah, I'm sorry. Give me a minute here. I'm trying to multitask here. So how was everybody's weekend? You guys have a good weekend? All right, still multitasking. Yep, excellent. I'm really glad to hear that. I saw a lot of you who hadn't finished your mystery powder labs finished them this weekend. So that was really awesome. That was awesome of you to do that. It was also, there were some pretty days, right? Did you guys get outside, soak some sun? You know, it's good to soak up some sun anytime you can. Almost there, guys. All right.
You have to take these if you don't have some.
Okay. Okay. Hang on just a second. Oh, gosh. So, do you guys have pets at home? How many of you have a pet? No? Yes? No? Some of you do, some of you don't, huh? What's your favorite type of pet? Do you like cats or dogs? We have cat or dog people in here. Dogs, both. Cats, both. Yeah, is that right? Can't make up your mind, like them both? I like them both as well, but my family is a bit allergic to cats, so we have a dog. But I do like dogs. I love dogs a lot. I go to the dog park a lot with my dog, and we have a good time. Okay, got that All right, so let's talk about this test that you guys took. So I'm just gonna. Oh, are you back, Rob?
Well,
Everything all right?
Yeah. Some took the retake version.
I don't think it matters.
Okay, then if they need to redo it later, yeah. I got him in.
Let him do it. Let him do it.
What is the dog talk about?
Yeah, well, I was asking them what their favorite kind of was, whether dogs or cats, and we have, we have some that like dogs, some like cats, and some like both. Yeah,
Good,
so I would say I like both too, but we have allergies, so we tend to go for dogs.
I like kitties and dogs.
Love cats. I wish we could have a cat. We had the greatest cat of all time, and he died. He had a long life, but he died. That's what happens.
Right.
Pets don't live as long as we do. It's really depressing.
Yeah, isn't it like every 7 years is 1 year?
Yeah, something like that. Yeah.
My dog lived to be 18, and I had a cat that lived to be 24.
Mm.
It was the bane of my mother's existence at the end. That thing was holding on for life.
I'll bet. I'll bet.
She's like, God, this thing is killing me.
Oh wow, that's a nice ripe old age. We had a breed that really wasn't. We knew it wasn't going to live past 12 years because the Great Pyrenees don't live much past that. But we had him. We had him for a good 11 years. Adored him. Okay, I'm going to share my screen, so I won't be able to see your chats anymore. But we're going to look at your item analysis.
I'll monitor the chat for us.
Okay, so we should be accordioning down here.
If you guys have a question, you can still put it in the chat. I'll monitor it. Miss Murdoch brings up the other thing.
There we go. You should see that, right? Can everyone see that?
Yes.
Okay. So here's question number 1, and it looks like a lot of you got this one right, so that's awesome. Unique properties of water enable life to exist on Earth. Which of these is a property of pure water? Okay, so the reason why D is right, it dissolves many substances. That's another way of saying it's a universal solvent, right? And that was one of the properties that we talked about. In fact, I talked about that a lot. I talked about how good water is at dissolving salt. Remember the salt ions that go into it, and anything can dissolve in water. All these other ones are trying to trick you because these other ones are not correct. They're the opposite of the properties of water that we learned. Solid water is not more dense than liquid. It's less dense. That's why ice floats. It has a low heat absorption? No, it has a high heat absorption. It takes a lot of energy to get water hot. That's one of the reasons it's nice to have all of our oceans and ponds and things made of water and not another liquid substance that heats up too fast. Or we would have boiled fish. It is slightly more acidic than air is a meaningless statement. I don't know who made that up, but they were not thinking very clearly there. Okay, next one. Enzymes only work with specific substrates because an enzyme, and its A is the best, has an activation site that fits with a specific substrate. I wish they would just call it active site and keep it that way because we called it active site and the textbook calls it an active site. Okay, somebody needed to say activation here. It's the same thing, okay? So it has an active site. Remember the Pac-Man and the Amoeba sisters? The mouth of the Pac-Man is like the active site, and that's where the substrate fits. And enzymes are really picky. Remember Mr. Bouchard made a big song and dance about that? So that's why A is the best, because enzymes have an active site that fit a specific substrate. All these other things are just distractors. They're trying to trick you. They're not right. Which of the following macromolecules are prominent? That means important. Okay, prominent means important. So which of the following is an important part of animal tissues that function in insulation, helping animals conserve heat? right? But which one of these will keep you warm? And the answer is lipids, because lipids includes our fat layers, right? So our fat layers, or the blubber on a whale or on a seal, those fat layers are there to help keep animals warm, and that's in the lipid group. Carbohydrates aren't going to keep you warm. They might secondarily keep you warm because your body will metabolize glucose, right, to generate a bit of heat. Your body does, but a layer of insulation on your body is fat, and that is a lipid. And if you had your macromolecule graphic organizer out, which I really suggest that you have the second time you try this, all of the information in these questions, almost everything, could be taken right from that sheet that we did together. So I encourage you to have that out. Okay. Question 4, both lipids and carbohydrates are important in animal cells because both, and the answer is they both store energy, right? Now, again, you can eliminate these other ones if you just had that graphic organizer out. You know both lipids and carbohydrates have CHO only. They don't have nitrogen, so B is wrong. Forming cell walls, no, that's not true. There's only one type of carbohydrate in hundreds, cellulose, that actually forms cell walls. So lipids and carbohydrates don't all form cell walls. Provide insulation? No, carbohydrates don't provide any insulation. We don't have a carb layer. You know, you eat chocolate cake, it doesn't form a sugar layer, a crystalline sugar layer on your body. No, it forms a fat layer on your body. So lipids are for insulation, but not carbs. Okay. This next question has been stumping students for years, and I still don't quite understand why. Maybe you guys just didn't play with blocks enough when you were little. This is just giving you shapes. Somebody laugh.
They say it looks confusing.
Okay, all right. Well, let's just look at this. This is a monosaccharide. They're just giving you a shape for it, right? So it's a simple sugar. This is a nucleotide. These are all monomers of the molecules we learned about. These are all monomers. This is a fatty acid. Here's an amino acid. And again, if you had your graphic organizer out, this should not have been difficult. So now if you look at the shapes here, you're looking for which diagram best represents an enzyme molecule. So an enzyme, you know, is made of protein. So which one of these is the monomer for protein? Enzymes are made of protein. So to make a protein, you have to string together a bunch of amino acids. That's all. So amino acids strung together is what you're looking for, and here it is. Here it is in A, right? So if each one of these is a monomer, they're giving you a nice picture of it, and they're looking for an enzyme, and an enzyme is a type of protein, and you know protein monomers are amino acids, so there's your answer. Okay. Next one. Question 6. I'm going to try to go faster through these. Here's an atom, right? Oh, you got 100% on this. Look at this.
Nice.
This diagram is to answer the following. An atom's nucleus contains, and if you look up here, here's protons and neutrons. So all you had to do really was read it. And there you go, both protons and neutrons. Right? Let's look at the next one. Okay, so this is a graph. Oh, and look how many of you got this right. This is awesome. All right. This graph shows, so let's look at it. This is looking at pH down here, right? So we're going from 0 pH to 7 to 14. And if you recall, this is where pH is low. That's where it's acidic. And when it's higher, that's when it's basic. And then this axis is measuring rate of enzyme action. So it's telling you how well the enzyme is working, right, on this axis. So pepsin is working best right there. Pepsin's working really, really well at that pH right there. And trypsin is working at a different pH. It has a different best pH. So it works best up here at this pH here. So now that's why the choice is, that B, pepsin and trypsin have different optimal pHs. Optimal means best. That's a good word to learn. So optimal means best. And you can eliminate the others because it says the enzyme trypsin works best at lower pHs. No, trypsin works best at the higher pHs, see? The enzyme pepsin works at pH of 9. Pepsin? No, pepsin works best at 3. The higher the pH, the more enzymes. It's not telling you how many enzymes there are. It's telling you how fast they're working. So D is trying to confuse you. So don't listen to D. Okay, let's keep going. Here's another atom one. Question 8. Here's a carbon atom. So how many covalent bonds can a carbon atom form with other atoms? Well, because in its outermost circle, it has 4 electrons on it, and they're lonely, they're unpaired. So because carbon has those 4 lonely electrons on there, it can form 4, choice D, 4 bonds with other atoms. That's really, really important capability of carbon. 9, which substance is inorganic? Inorganic, meaning doesn't have carbon or hydrogen bonds in it. So you should know that water is the inorganic thing in this list. And good for you if you chose that. All these other things, glucose and glycogen and lactase, those are all organic molecules that we've talked about in your macromolecule organizer. Okay, here's another way of looking at inorganic and organic. Here's a table. Groups A and B contain molecular formulas of compounds. How would the compounds in these groups be chemically classified, or how would you organize them? So the correct answer is C. Group A is organic, right? These are all organic. Look at the carbon and hydrogen. And group B is inorganic. No carbon and no carbon hydrogen. This C here is part of chlorine. It's not a carbon. So this is a single ion of chlorine. So don't confuse it with carbon. Okay, most of you got that right, so good for you. Okay. This diagram seems to have confused people. So question 11. The diagram below shows a portion of a nucleotide, right? So a nucleotide, which substance could complete a nucleotide? So remember, a nucleotide is a phosphate, sugar, and a base. Here's the phosphate. Here's the sugar. This is some sort of a base that needs to go here. So again, you had a macromolecule organizer that had this exact picture on it. And you had notes that also talked about this. So there are 4 different bases that can go here. Adenine, thymine, cytosine, or guanine. Those 4, A, T, C, or G, goes here, right? And adenine is the A. Adenine is A. So that's the only one that could go there. These other choices are entire molecules that have nothing to do with a nucleotide, right? So a protein is not any part of a nucleotide or a nucleic acid. An amino acid is part of protein. So those two are right out right there. They have nothing to do with this. RNA is an entire nucleotide that's made of these guys linked together. So RNA is a larger molecule made of nucleotides, so that doesn't go there either, okay? So RNA is a string of these. Adenine is the only answer there.
Yeah. Hey guys, when we're taking these tests, you got to use some test-taking strategies. Because not necessarily is it going to be just, is this it, this it. Sometimes you're going to have to look at how the question's worded. If you don't know the necessary answer, maybe you're like, oh, I don't even know. I'm just going to pick a number. Why don't you look at it and cross out what you know it's not, and then narrow it down? Because, you know, if you knew it wasn't RNA and you knew it wasn't an amino acid, you could have gotten 50-50 as opposed to just giving up right away. And a lot of these things you got to look at closely because these test questions are taken right from the SOL. And SOL is not necessarily about knowing specifics about biology. It's applying the concepts and understanding of biology to answer questions. And a lot of the questions are graphs and pictorials. So that's why we're giving these to you now, so you can start seeing these and be aware of how you got to think when you take the tests. Okay, so We're going to have a lot of graphs, and our goal is to understand graphs and pictorial representations. Those are never going to go away. They're going to be, I still look at those now in my meetings.
Yeah.
So look at it, read through it, try to answer before you even look at the answer, see if you know it in your head. And then if you have it down there, you might be already there looking at it. Just another thing to think about, guys. But take your time and look through and cross out what you know it's not. Sorry, Ms. Murdoch.
No, that's great. That's very good advice. Elimination techniques, that's what that's called, right? So if you're not sure what the answer is, cross out what you're pretty sure it isn't. And I think you have a strikeout option on your computer that's allowed. So you can actually click on a strikeout and like line something out. If you're sure it's not that one, then you don't have to let your mind be distracted by it anymore. Okay. Which compound has this structural formula? So this, we showed you this lots of times, and this is also on that graphic organizer. It's right there, and I pointed to it, and I even named it. This is glucose. This is glucose, and you're absolutely sure that it's glucose because it's got that hexagon structure with the oxygen as part of the ring, which fructose doesn't have, by the way. Okay. Fructose is the only other one that I might have thought, I understand why they would have guessed that, because it has the exact same number of carbons, hydrogens, and oxygens. But no, this is glucose, and we saw this structure lots of times. So that's a really good one to be able to recognize. Okay, on with this one. This is straight out of the class notes that I gave you one day. I actually drew this exact picture for you. So base your answers on this diagram, which represents stages in the digestion of starch, and on your knowledge of biology. Okay, so the products, we talked about reactants and products, right? So here's an enzyme, and here's its substrate starch, and look how well it fits in there. And then after the enzyme is done with the starch, it breaks into its parts. And those parts are going to be simple sugars. Because starch and simple sugars, first of all, they're in the same family of carbohydrates. So it's really the only thing that you could break a starch into is smaller sugars. These other things, fats, amino acids, minerals, these have nothing to do with carbohydrates. Starch is a carbohydrate. A fat is a lipid. If you break a starch into its parts, you're not going to get fats. You're not going to get amino acids. Minerals is right out. I don't even know why that's there. But that again, that's why I wanted to do that graphic organizer with you, because literally it keeps your mind from making those organizational mistakes.
It was kind of worded weirdly. What do you mean? That's a good question. I need to know what you mean. Like, based on which represents the stages in the digestion of a starch on your knowledge of biology. So when they're asking about digestion, think about that word. Think about the word. So the key in there is digestion. When you're digesting, what are you doing? Are you building or breaking down? So the product, would it be, you're not fats, no. Amino acids and minerals, no. You're probably breaking down to simple sugars from more complex sugars. So you got to think of it like that. It's not necessarily going to be.
Starch is your reactant. We talked about that, right? Starch is your reactant, and then there's an arrow, and then products on the other side. So the products have to be parts of the original reactant that just got reorganized, right? The products have to be just smaller bits of the reactant. So you're taking starch and you're breaking it down. And digestion is, that's just another way of saying breaking down, because that's what we do when we digest something. If you digest food in your stomach, you're breaking it into smaller bits. I hope that helps. Okay, same picture again, except this time I asked you to identify what X is, right? So this one, yeah, I see why. Okay, so here's another test taking technique, Mr. Bouchard, you should tell them about.
All right, using this diagram.
Okay, but look at what their answers were.
Oh yeah, no, an enzyme. Okay, so the identified molecule X is most likely. And which one? All of these are correct. Okay, here's what I want to talk about.
Yeah, you see what I mean?
Guys, so what you want to do, sorry, I had to figure out what you want to do. You want to look at that, okay? You know this is an enzyme. What are enzymes? Think about what you know about enzymes. Enzymes are a catalyst as well. They help speed up and slow down reaction. And enzymes are also a protein. So every one of those things, see what you know about this. If it's an enzyme, you go, okay, it's an enzyme. Let me see the other answers. Oh, enzymes are proteins. Proteins, enzyme. So it's got to be a protein. So you have two of them. And then even if you have two of them, and then you know it's a catalyst, even guys, you can already go down. All of these are correct. So what you want to do is look through it. You gotta. A lot of you guys are just looking through probably the top two options, and not even going, because once you see the answer, you're like, that's it. I know it's an enzyme. I know it's an enzyme.
And you're right. But there's
right.
other ways to describe an enzyme.
And all three of those are other ways. So I guarantee you, the people that got it wrong, and I love you to death, you probably looked at it and go, I know that's an enzyme. Enzyme, I'll put an enzyme. And you're right.
Right.
But
it's also a protein, and it's also a catalyst.
Yeah.
Just read all the choices. Read all the choices. That's it. Next, 15. Let's go. All right, so now we have, and this I actually drew for you. I know I did. I drew this for you on the whiteboard.
It's in the
glucose and glucose, and I actually drew this exact same thing. Remember this? And this is dehydration synthesis, right? You're losing water and you're making maltose because you've linked those two glucoses together to make a new molecule. So that's a dehydration synthesis. Hydrolysis, you may have gotten confused because water is involved, but hydrolysis means to break with water, not to build with water. So dehydration synthesis is putting together, and that's what we did. Hydrolysis is opposite, taking apart.
And guys, when you see these pictures, if you see an arrow pointing out with a thing, that's something that's leaving. It's not being added. That's the way you also got to be aware of. Water is
another product. Water is another product, and I made a big deal of that when I did that whiteboard thing. So if you're watching that class before, when I was reviewing with you, I did this question right there on the screen for you. So these are the two products. The diagram is showing a chemical reaction. Which of the following represents the correct products? You see, I actually gave you this answer before the test. So what are the products? Maltose and water. Maltose and water. There you go. Maltose is not the only product. I actually told you that before you took the test. Okay, so be careful of that. All right, here I gave you a graph. Oh, and good for you. So many of you got this right. This is awesome. I actually wrote this question myself. Which of the following is a correct conclusion to make from this graph? Okay, so if you look at it, the first choice is the activation energy is greater for a reaction if the enzyme is present. So let's look. Oh, here it says activation energy, and there's this arrow here. And this one is another arrow that's that. So there's a shorter one. The shorter arrow has with enzyme. Okay, so what that tells me is that A is not true, because activation energy is not greater when the enzyme is present. It looks like it's greater without it, without the enzyme. The activation energy is greater if it's not present. So good for you if you guys looked at the diagram and just allowed it to tell you what the answer was. This is a really good example of, you know what, if you've got good reading skills and graph image processing skills, you don't even need to know what activation energy is to answer this question. You can just look at the wording and the size of those arrows and figure it out. And sometimes the science is like that. So trust your visual and reading skills. And again, if you have a word that you're not sure what it means, come and ask us, okay? Okay, here's question 18. The big, long molecule, and I wrote this one too, and I had trouble getting this smaller, so there it is. It's this big, long molecule. And it says this molecule is a blank and can be found blank. Okay, so this is a phospholipid, and you can tell that it's a phospholipid because it has a phosphate up here at one end, and it has lots of non-polar, it has these big, long, non-polar tails. It even looks like a phospholipid. If you've seen cell membranes, there's the phospholipid. I also gave you it as a picture. So that's why A is correct, and it's found in cell membranes. Okay. This is definitely not DNA. DNA is a twisted ladder, and that does not look like a twisted ladder to me. So definitely not. Okay. All right. Which of the following, oh, got this right, that's awesome, is an example of a protein with its correct function? All right. So C, actin and myosin form structure of muscle fibers. Very good. And this was straight out of the notes as well. Guys, I gave you these. Hemoglobin is not part of animal hair. No, hemoglobin transports oxygen. So I reversed these. I wrote this one too. Keratin transports oxygen in the bloodstream. No, that's hemoglobin, right? Enzymes do not store gene information. Enzymes speed up chemical reactions. So that's not right. So there, that's why C is the answer. So good for you if you got that right. 20. This is a twisted ladder. This is DNA. Okay. So use this figure to answer the following. This molecule is found in blank, and its function is to blank. So this molecule, you're thinking it's in the nucleus, maybe, is what you're thinking. But I was a little more specific, and I said, oh, it's in the chromosomes, right? C, it's in the chromosomes, and it stores genetic information. So let's see how you did with that. You mostly got that one right. Okay. A student is testing an unknown solution. Here's the chemical indicator lab, guys. So if you did this, you got this right. And if you didn't do it, you probably had a real hard time with it. So we're testing for glucose and starch. Benedict's solution, and heats it, and the color is blue. So Benedict's tests for glucose, and if you heat it and it stays blue, glucose isn't there. When she adds iodine, the color is black. So iodine tests for starch, and black is a positive color. So we've got no glucose, and we do have starch. So here we go. Starch is there, glucose is not. So the only way you could have gotten that is if you really paid attention to our big chemical indicator lab. That was the big lab. And if you, even if you didn't, you probably mostly know that adding iodine and it turning black, no, that's a starch indicator. So you might have gotten it just by elimination. But yeah, Benedict's is already blue. So if it stays blue, there's no glucose there. That's why A is wrong. Okay. Is it dangerous for a person to have a high fever because and you did better on this one, because all of these happen. Yes, everything. Enzymes vital to life can denature. Active sites on enzymes no longer fit their substrates. Enzymes can no longer lower activation energy of vital reactions. Those are all right. Okay. And here's water molecules. So this looks like some kind of tangled, like, little thing that you may have built with your Tinker Toys when you were a kid. But what you're looking at here is separate water molecules. Here's one. Here's another. Right? I wish they had color-coded this a little bit. There's another. There's separate water molecules, and they're connected by these dotted lines. And I drew this for you a couple times. I was showing you that the dotted lines are weak bonds. They're called hydrogen bonds that sort of lightly connect water molecules together, and this is responsible for their cohesion. For cohesion, which means water molecules stick to each other. Adhesion is different. Adhesion is water molecules sticking to other objects, like the inside of a test tube or the inside of a vein in a plant. So adhesion, water sticks to other things. Cohesion, it sticks to itself. So that's why B was correct there. Which of the following nucleic acids acts as an energy transferring molecule? So that's ATP, is about energy. These other ones, all of these other ones, DNA and RNA, any type of RNA, are involved with genetic information. DNA and RNA are never going to give the cell energy. That is not what they do. ATP is the only one of these that will give the cell energy. So now you know. And this thing, this is a fat molecule. This is a triglyceride into all its glory, okay? And it's saturated. There's not a single double bond anywhere. There's no kinks, nothing. This is going to be found in animal fat, fats that are solid at room temperature, which animals like because otherwise we'd be sloshing around like Baba Papas, right? Okay, so you would find this D in the blubber of a whale or fat layers of a human because it's saturated, it's animal fat. Peanut oil is a plant oil, and plants tend to have liquid fats. So they can only be liquid if there's a kink, an unsaturated fatty acid in them. So if there was a bend here because of the double bond, then you'd have oil. And it's not a phospholipid. Phospholipid was in a different question, and there was a phosphate over here, right? A PO4 group over there. So it's not in the structure of a cell membrane. And I think that's the end. There we go.
All right. So what you guys need to do right now is log on and take the test, the retake. It is entitled. I'm right back in my test bank here. Hold on.
It says retake in big letters.
Yeah.
Should be pretty easy to find. And get all your notes out and take your time.
Okay? That's what you should be doing right now. And some of you did it while we were reviewing here.
Yeah.
So I'm going to.
Yeah. If you've done that, then we need it's a short break, and then.
Yeah.
And
Go ahead and get on there. I made it unavailable for third period, Miss Murdoch, because I want them to pay attention to the review before people.
Okay. Sure. That's fine.
I was like, I'll make it available once you're done. I just think listening to us is important.
Okay.
So go on. If there's questions, let me know. I'll be in there in the system. I'm in the system now. Go to test bank.
Mm-hmm. And if you are all done, are you going to send them to the testing room then?
Do you want me to send them to all of the tests? Well, they can go there automatically on their own.
Yeah, let's have them go there. And if you are all done, stay here, and I'll send you into something else. Okay? I'll talk to you about something else.
I'll be in there. All right.
Okay. Okay, guys, that's good. I just need an answer. Just let me know. There's no pressure. I just want to know who's done and can go on. Okay, that's okay. Okay. Okay, so if you need to take your retake, you should move to the other room. If you're able to move, you should be able to move. So you can take it there, and then if you're done, stay here, and we'll talk about the algae lab. Okay. Go ahead and join test makeup and take your time in that quiet room to finish your test, to do your retake. And if you're all finished, just stay here with me, and I'll show you what to do. Okay, so if you are done with your retake and you're happy with everything and everything's fine, this is what we're doing now, the algae lab graph and claim evidence reasoning. So if you go into your classwork tab, excuse me, I didn't eat breakfast yet. You go down to where it says, here, let me go page. You go down below the unit testing review, you're done with that. Below biochemistry, you're done with that. And then there's graphing assessments. And right at the top is algae lab graph and CER. So go ahead and look at Mrs. Murdoch Explains How. Okay, just open that. It's not an Edpuzzle, it's just a YouTube. If you haven't already looked at it, go in and look at that. And then open your algae lab spreadsheet, doing what I explained how to do in the video. Somebody just chat me. Oh, Sakira, that's awesome. That's great. Okay. Okay. Okay, so if you are in this room and you're still doing your retake, I would appreciate it if you would move to where Mr. Bouchard is, because I'm going to talk about the algae spreadsheet in the lab right now. So I think there are many of you who are done with your retake and can handle that information. So if so, great, stay here with me. But if you're retaking and you don't want to have me talking over your retake, move to Mr. Bouchard's room. Okay, so if you're here and you're ready to do this algae lab, like some of you are, oh, that's okay, Ethan. That's why I did the video. Okay, so in case you lose me, that's fine. Go right, do what you need to do. Okay. Okay. So let me just share with you this. I mean, this is where we are right now. This is your Google Classroom, and you go down to graphing assessments, which is the topic beneath biochemistry. And there's algae lab graph and CER in there. And some of you I know are already looking at the video, and that's great. But now I'm live and I can talk to you live, and this is the last time I'm going to get you live until next Tuesday. So I'd like to take advantage of that. So whatever you're doing, just stop so I can help. So I can just give you some information and we can interact directly so that you can at least get the graph done by the next class, because you guys are smart. You can do that. So let me share with you. I'm going to stop sharing this. You should know by now where to find this, and you should be there, okay? I'm going to just share, let's see. Yeah, I'm going to go to my copy of my, there we go. So you should be able to see on the screen. Yeah, come on. There we go. Okay, so copy of algae lab spreadsheet. So this is what you're going to play with, and you're going to make a graph from this. And I've already put all the data in here. You don't have to add anything. It's already here. So you guys have been taking your data onto your own worksheet, and that's good. I'm glad that you did that, because that way you felt involved with this. And now we're going to use the spreadsheet to construct a graph. But we've got to do some calculations first. So my video already kind of went through this. I don't want to bore you. But you're going to put some calculations here in the boxes I'm highlighting here. You're going to calculate total change in the growth of the algae per cup. So we had 8 cups. So there's these 8 boxes where we're going to calculate the difference between where the cup started, like 0 milliliters, your control A cup started at 82% and went all the way down to 27.6%. And you want to calculate the difference between these two numbers, between 82 and 27. And the spreadsheet will do that for you. I mean, you could just use a calculator and do it that way, but the spreadsheet does it for you. If you type an equal sign, and the computer is waiting for you to tell it what to put in there. Now, I could just go 82 minus 27.6. I could just do that, and it would do it for me, right? It does it for me. But I'd like to show you a little trick that makes it easier. You can actually go equals and go, okay, I want this cell minus this cell. You just click on the cells and then it does it for you. It's the same answer. Not only that, but if you take your arrow and you move it to the right-hand corner, see how it turns into a black X like that? You can drag it across and it will do all of them for you, just like that, really fast. Now that all of these numbers are differences, right? They're a subtraction between this top number, whatever it was, and this bottom number, which should give you this. And see, if you check all your answers, they should be right. So these are the total changes that happened over those 2 weeks between October 7th and October 16th, or a week and a half. Sort of, right? And we felt that Mr. Bouchard and I were looking at the cups and we're like, you know, they're evaporating, the volume was going down, and we felt like it had gotten to a good place to stop it. So we decided to stop it there, which is fine. So now you have to graph this. And what you're going to do is you're going to combine your, but before we graph it, the other thing is that you have to do some more data crunching, because this number here, remember, was a 0 milliliter, but that was your A cup, right? And then this number over here was also a control, right? It was a 0 milliliters of nitrogen, but that was a second cup. So we did duplicates. We did 2 of every type of cup. And now what we need to do is take the average of the difference that happened in those duplicate cups. Let me make sure that that's right. Yeah. Oh, no, it isn't right. There. So there's your 0 milliliter B cup there. So this number and this number, we want them to be pretty consistent. We would like there to be not a whole lot of variation there because we did the exact same thing to those 2 cups. We didn't add any nitrogen, and then we measured them every day to see how much the algae grew, right? So we need to take an average. So once again, we're going to go down here, and we're going to calculate an average in these places. And this sets up the data really nicely for your graph. So the 0 milliliter, there were 2 of them. There were 2 of those cups. This was one. See, I labeled it here. That was one. And that's the other one. And I want to calculate an average of them in that box. So I'm going to go equals, and I'm just going to very simply do, you know how to do an average of something. You add them together and divide by 2, right? So I'm going to go parentheses, this number plus this number, close parentheses, divide by 2, right? Which gives me an average there. Okay, and then for the others, I'm going to do the same thing. So it's going to be, this was your 5 milliliter, and I'm just retyping it so I can see it better, right? This was your 5 milliliter B, right? So those 2. Here, so I would go equals 5 milliliter A cup. Oh, parentheses first, otherwise it doesn't. 1 plus 1 parentheses divided by 2, and it does it for you. Right? So you fill those in. Okay, you fill those in, and then you want to do a graph, right? So I'm just going to do part of the graph just to show you quickly how to do a graph. You should remember how we've done graphs before. You did the homework in graph. And you know, before I do that, I just want to go back here and check that you're all here. Could you all just give me a thumbs up? Oh, Pedro, are you having trouble with that? Maybe I can try. Yes, thank you. Could you just let me know if you're listening? Because I feel like I'm talking to a screen. Oh, great. Thank you. I'm really glad. So what you're seeing me do exactly what I did in the video, aren't you? If you watch the video, which is fine. Okay. I'll just show you again. I just want to make sure. Please don't feel like you can't ask me something, okay? If you want to put your mic on. So Pedro, I'm not sure how to help you with a calculation issue, but I mean, like if I just put my if I identify a box like that, if I click on one of the cells, I click on a cell, right? And then I go equal sign. If I don't press equal sign, it's not going to calculate anything for me. So maybe that's the issue. Type an equal sign first, and then the computer is waiting for you to tell it what to do next. What do you want to calculate for you? That's what it's asking for. Did that help? Pedro, did that help you? Okay, so it's working.
I'll try it again. But
Okay.
for some reason, when I put the equal sign, it still doesn't work when I click on it.
Okay, are you in the spreadsheet? You're in the spreadsheet, same one I'm in, right?
Yes.
Okay. Oh, are we at the end of class? I thought we went to 11:35. Mr. Bouchard, am I going over? Oh, I don't know. Listen, I'm gonna stay with you to make sure that you can calculate stuff. If you want to stay after with me with calculation issues, I just want to quickly show you how to graph this, okay? And it's looking like we're not going to be able to really finish this until we meet next week. So this will not be due Monday, all right? This will be due probably the end of next week because we still need to talk about it. And there were too many people who needed to finish their test today, okay? So I'm not going to stress you out about that. But if I type an equal sign, it should go, you know, if I go 2 plus 4, and then, you know, it should do it. The only reason I can think it wouldn't is if maybe you're trying to go on top of something that something was already in, or you're not all the way back, the cursor isn't all the way to the left. Keep trying those things. But to make a graph really quickly, you're going to highlight the X and Y descriptions, go down and highlight the data. I'm just going to make a really short graph here, right? And then I'm going to go insert and chart. And you do want a bar graph for this, so you can just leave it as a column chart. That's fine. And then to customize it, you know that you need to put chart and axes titles on it to get full credit. And this isn't finished because I didn't finish my data yet, but I just wanted to show you how to do it. So there's for your chart title. There's for your horizontal axis, which would be what? What would your horizontal axis be? It should be contents of the cups, right? Or concentration of nitrogen in the cups. That's what it should be. And then your vertical axis should be percent transmittance or change in percent transmittance, which is what it says, right? Top of there. So it should already be there. But that's how you do that. And I think we are at the end of class. Yeah. Yes, I'm still here though. Yes, Pedro.
So when is this due?
Like I said, it's not going to be due till the end of next week because we still have to catch other people up with this. So I'm going to change the due date, okay?
Still doesn't work like
Sure,
this.
yeah, because you guys all had to do your tests today, so that's fine. A lot of people had to do that, so we just didn't have time to get to this. But if you're already in this, you can go in there and use my video, play with it, see what you can come up with for a claim, evidence, reasoning, but don't turn it in until we've talked about it together, okay? So you can do your best work. Does that answer everyone's question? I hope so. Okay. I hope you have a good day. And if you are having calculation issues, come to my office hours on Monday, and I'll try to help you. Okay? But not due until I'm going to change the due date, so don't worry about that. Okay? So Pedro, did it get resolved or?
Oh, no, I still can't get it to work. Like, I write the equal sign, and then when I click on like a cell or like a number, it just like does as if I wanted to write in it, but it doesn't necessarily like write it down in the.
Oh, okay. I don't think I've ever heard that problem before. So like if I go here, and let me just get rid of this here. Like if I go into a cell here, right? And I
Yes,
go equals and I do that. Plus,
it doesn't write it down. It just
Really?
like does as if you wanted to write in that box. Yeah.
does it let you type something?
Yes, and the box that I click after, but it doesn't like put the number in the.
So is it not doing that for you?
No, it doesn't do that.
Ah, that is really weird. And you're using the Google spreadsheet, right?
Yes.
I don't know. That is a really strange thing. I mean, Pedro, you can do this with a hand calculator if it comes to it.
Yes, of course.
You know, I mean, you can do that. It's not going to change. The more serious thing is if it doesn't let you graph something, right? I hope it won't do that to you. But I mean, you can just do it with a hand calculator for now. I don't know why it wouldn't be letting you do that. I'm sorry.
Okay. Thank you. Have a good day.
Yeah, have a good day. Have a good day. Bye-bye.